I have a simple question for you. Why are you alive? While this might sound philosophical, the answer is pretty straightforward. Because the human race evolved. According to Darwin's theory of evolution by natural selection, organisms better suited to survive in their environments tend to live long enough to reproduce. Their offspring inherit these advantageous traits, passing them forward to future generations. Conversely, organisms that lack such beneficial traits fail to reach maturity or produce fewer offspring, and their lineage gradually diminishes. The main takeaway is that humans and many other successful species survived and thrived because we possess favorable traits that conferred an evolutionary edge. Fast forward to the 1990s, Dr. Frances Arnold took these Darwinian concepts and brought them into the lab, pioneering the field of directed evolution. The approach begins with a naturally occurring enzyme and its corresponding gene. Researchers mutate this gene in various ways and reinsert their modified versions into bacteria cells, which then produce many enzyme variants. From there, scientists apply a selective pressure test, such as improved thermostability or better antibiotic resistance, to identify which variants perform the best. Through iterative rounds of mutation and screening, they gradually refine the enzyme until it reaches desired levels of performance. However, early directed evolution methods often relied on randomly mutating parts of the protein, which wasn't always optimal. Enter a new era of targeted mutagenesis with a technology called EvolvR. It integrates the CRISPR-Cas9 system with an error-prone DNA polymerase. Recall that the CRISPR-Cas9 system is adept at targeting specific locations in the genome. It does this using a spacer, a short sequence that guides Cas9 to a specific 20-base target region. Once the Cas9 binds to this region, it acts as a molecular scissor, creating a small nick in the DNA strand. This is where the error-prone polymerase comes into play. A DNA polymerase's job is to synthesize complementary DNA strands, but an error-prone polymerase does so poorly, introducing a higher frequency of mutations as it rebuilds the strand in the 3' direction. Meanwhile, cellular enzymes chew back the 5' flap and then repair the DNA with these introduced mutations now incorporated. This leaves us with a new mutated sequence at a targeted location, one that can be iteratively improved, much like Darwinian evolution, but with much more control and speed in a lab setting. This approach not only refines the directed part of directed evolution, but it also expands the toolbox for researchers to continue optimizing and generating new variants of enzymes. Thank you. I hope you enjoyed this talk.